please. He's going to present his work on adversarial domain adaptation. And here's the pointer.
Good morning, everyone. I'm Taylor from Carnegie Mellon. It's my great honor to be here to present our work on adversarial domain adaptation framework for cross-data source macromolecule classification in cellular electron cryo-tomograms. This is a joint work with Xiangrui Zeng, Dr. Chris Kitani, and my advisor, Dr. Ming Xu. Our research focuses on macromolecules. Macromolecules are involved in many cellular processes. To fully understand these processes, it's necessary to know the structure and native spatial distribution of the macromolecules inside single cells. There are many types of macromolecules, each with a different, as shown here, each with a different structure for different biochemical function. The information of macromolecules' native structure and spatial organization has been very difficult to obtain. The recent advances in cellular electron cryo-tomography has made the acquisition of such information possible. This is an in situ 3D imaging technique which images subcellular structures through tooth scaries, which are later to be reconstructed to 3D tomograms. Since the data is in their native environments, it's very important to detect and identify the structures inside, especially macromolecules, for downstream analysis and spatial modeling. This is a slice of a tomogram. Going from the left figure to the right figure is the process of structure pattern mining, sometimes also called visual proteomics. Given a reconstructed 3D tomogram, potential structure regions can be extracted as subtomograms using structure detection methods. Subtomograms are small cubic subvolumes that generally enclose one macromolecule. These are the protein databank ID. To identify and classify the macromolecules inside a subtomogram, researchers have proposed many methods in recent years. Since 2017, deep learning-based subtomogram classification methods has been proposed and attracted a lot of attention. In the SHREC 2019 track, classification in cryo-electron tomograms, organized by Dr. Friedrich Foster, 8 groups from 4 countries participated in the competition. Based on the results, the track concluded that learning-based methods are increasingly more popular with cryo-ET researchers, not without a reason. The learning-based methods show better performance, especially with 3D convolution. By the way, our lab achieved a tied best performance on macromolecule classification, macromolecule localization, and a second best performance on macromolecule classification. Nevertheless, different data sets have different image intensity distributions due to different experimental conditions. When applying a trained classification model on one dataset to a new dataset with different imaging conditions, the accuracy is usually very low. This phenomenon is termed domain shift in cross-data source prediction. Practically, part of a new dataset must be annotated to predict on the rest of them, but the computation and manual quality control cost of the annotation can be very expensive. Therefore, it's ideal to transfer the knowledge from datasets, simulated datasets, or datasets already annotated to new datasets. Formally, there are three types of domain shift. The first one is covariate shift. It's a case when the conditional probability of the class label given the subtomogram data remains, but the covariate of feature distribution of the subtomogram data changed. The second one is prior probability shift. It's a case when the prior probability of the class label changes. For example, maybe 10% of the subtomograms in the source domain belongs to class 1, whereas 80% of the subtomograms in the target domain belongs to class 1. And the third one, concept shift. Concept shift is a case when the conditional probability of the class label given the data changes. For example, John Nash is classified as Nobel Prize winner in the source domain, but in the target domain, he may be classified as Carnegie Mellon graduate. And this is a pretty unlikely scenario for our task, because to be rigorous, a ribosome is always a ribosome in our data. To illustrate, we have two simulated data sets with different spectral aberration and defocus. As shown here, the same structure appears differently in different dataset batches, and also for this structure. Across all signal-to-noise ratios, it seems like dataset batch B has a slightly better contrast but their appearance difference in their features will lead to low classification accuracy when training the model on one dataset and predicting on the other. After surveying a few number of deep learning-based domain adaptation methods, we adapted an adversarial domain adaptation framework to our data. It has 3 stages. The first stage is just standard classification model training on the labeled source domain datasets. And after the first stage, the source feature extractor and classifier stay fixed. The second stage is to train a target feature extractor to be domain invariant in an adversarial training fashion, so that the target feature extractor during the training process tries to fool the domain discriminator so that the domain discriminator cannot tell whether the extracted features are from the source domain or the target domain. And during the third stage, since the target feature extracted are already invariant to domain, the classifier trained from stage 1 can be directly applied to the target feature extracted to get the target subtomogram labels predicted. We have 2 traditional domain adaptation methods baseline to compare. The idea of direct importance estimation is very simple. It tries to find a weighted feature distribution, target feature distribution, to be as close as possible to the source feature distribution in terms of maximizing the KL divergence. The second baseline is to learn a structural correspondence between the 2 domains and project them into a shared feature space. Then the training and testing of the model can be performed in that shared space. Here are the algorithm and model architectures of our method. The feature extractor is similar to what we have designed previously for subtomogram classification. And the structure classifier is trained simultaneously with the feature extractor. The output of the features will be directly inputted into the structure classifier. And the domain discriminator is just a sequence of fully connected layers with leaky ReLU activation. For testing, we simulated two dataset batches, A and B. Each contain 5 datasets with different signal-to-noise ratio. There are 23 classes of subtomograms in each dataset, as shown here. And there's also one null class with no macromolecules. This is basically all the macromolecules we're not interested in. And as I mentioned, the two datasets have different imaging conditions, including spherical aberration and defocus. This table shows the cross-domain prediction accuracy, and the model is trained on a source domain dataset from batch B and predicted on the target domain dataset from batch A. And in each cell, these are the SNRs, and in each cell, the first row shows the accuracy without domain adaptation. The second row, direct importance estimation. The third row, structure correspondence learning, and the fourth row, our proposed 3D ADA framework. The highest accuracy in each cell is highlighted, and as you can see here, in 24 of the 25 cases, our proposed 3D ADA framework achieved the highest accuracy except only here. And it's close to the best one. Here are some visualization results of our results. And the TSNE embedding shows that after the domain adaptation, the features of different classes are better separated. And the confusion matrix shows that the classification accuracy significantly improves after 3D ADA as the diagonal darkens here. We also have 3 experimental datasets, each with 3 classes. And experimental datasets are very challenging to classify due to their low signal-to-noise ratio. Here are the 2D slices representation of some subtomograms example. And as you can see here, you can barely see a structure inside. Although denoising can sometimes help you with visualization, but for deep learning-based method, denoising actually decreases their prediction accuracy. It's probably because that many macromolecules look similar to each other, and denoising will also reduce some structure details. So we train the model on simulated datasets and predict on those experimental datasets. Without domain adaptation, the classification accuracy is close to random guess. And although domain adaptation significantly increased the accuracy, we still think that there's a large room to improve in the future. When the extracted features are better separated, unsupervised clustering can be applied to the extracted features to detect novel structures that does not exist in the training data. We have a novel structure, RNA polymerase here, and those red dots show that after domain adaptation, they are better clustered. Though here it does not look like a cluster here, but the TSNE embedding is just 2-dimensional. In higher dimensions, they are better separated. And here are the structure recovery results using subtomogram averaging. On the left are the structure templates, and on the right are the recovered structures. Only after domain adaptation, the novel structure can be successfully recovered from the data. And for other existing structures in the training data sets, the structure recovery also improves. To summarize, preparing training data annotated training data is a major bottleneck for subtomogram classification. It's beneficial to have separate training data source where the annotation is either readily available or can be performed in a high-throughput fashion, such as using simulated training data. And the new problem is that how to deal with domain shift. in cross-data source prediction. And our results shows that the proposed 3D ADA framework stably improves the cross-data source prediction under different imaging conditions. With that, I would like to thank you all for your attention. I would like to acknowledge the funding agencies for their support. Our lab website, GitHub, and my email are shown here, and the code is available on GitHub. Thank you very much. I'm happy to take questions.
Thank you. We have time for a few questions. Is there one? If not, I would have a question. It's not.
I think there's
really,
one question.
There's one question. I'm a bit blinded by the light. Is there one? Oh yeah, yes, please. Please go ahead.
Yeah, I was wondering, how does it handle rotations of the data if the structures are rotated? Is it still invariant to that?
Yeah, so that's a very good question. So for domain adaptation, we do not take rotation into consideration because there's no evidence showing that the rotational preference, there's a difference between the source data site and the target data site. But in terms of subtomogram classification, since the structure, the macromolecule structure is randomly rotated inside subtomogram, so that if the training data set has a diverse rotation for each structure, the testing data set will be fine. It can correctly predict the same structure even if they're in different orientation, also for small translation. But I think I think we could include some, like say, rotation invariance or rotation attention technique into the subtomogram classification methods.
Is there another question? In fact, I would have a question. A very alternative strategy of approaching the problem could be that you start labeling points from the target domain and train a classifier just on the target domain. And I think in practice it would be very, very interesting to know how many points do you have to label in the target domain until you have a sufficiently large training data set to outcompete, or to outperform the domain adaptation approach. Have you thought about this? Is this something you have on the to-do list?
It's a very good suggestion, and my idea is that the rationale for this method is to reduce the annotation cost, because it may take several weeks to annotate those, because you need to compare it with the template at all rotation and translation, and it's very computationally intensive. And usually, the training data set need to be, say, sufficiently large, maybe several times larger than the testing data set to have a satisfactory performance. And because of the noise, and also there's a missing wedge effect, like some image distortions.
And I understand. Of course, in the experiment you showed, you must have the labels on the target domain also, because you evaluated the accuracy, right? So in this setting, you could study this, but I see the point, you may need a large data set or a large number of labeled samples too.
Yeah, it took us a lot of time to annotate the training data set because it's not automated. After the template match process, we also need to do some manual quality control to visually look at them and see whether it's that structure or not. And it can be biased because different biologists may have different opinions.
Good, thank you. Oh yes, now there's one more question. We have exactly time for that question, yeah.
It's a really interesting talk. I mean, I think the general idea of using simulated data to train an algorithm and then domain adaptation to make it fit to the experimental data is a great approach and have lots of applications. Just one question. I mean, I've heard that training of adversarial methods can be kind of difficult, finicky. Did you have any difficulty with actually making the adversarial training work effectively?
Let's go back to this slide. Actually, the the difficulty is not, it's kind of small because, at least during our training, although there's domain shifts, they look more or less similar. The difference is smaller than the similarity, so that we have the source feature extractor fixed, and then we the target feature extractor will be a little bit similar in terms of their convolutional features, and we just want to try to modify a little bit the convolutional feature to make it as close as possible to the source feature extractor, to the source features. Yeah, so, and it's trained in an alternative fashion. We train the domain discriminator for one stage and fight it with the target feature extractor for one stage, then we train it again, and it will eventually come to a stable point where the domain discriminator cannot tell the source of the features.
Okay, thank you very much.
Thank you.
Thank you again to the speaker and to everyone who asked questions. Thank you.